Today we will discuss about the microbial genetics. Okay, so before that we will see like how the bacterial chromosome is and how it is means transferring from one bacteria to other bacteria. Okay, so bacteria typically it is a single cell. Okay, single it is kind of prokaryote, so it is having a single cellular organism which is having a single circular chromosome consisting of a single circular molecule of DNA which is associated with the proteins. Okay, and this means apart from its own bacterial chromosome, there is a kind of small extra chromosomal DNA is also there, that is called the plasmid, and this is the bacterial chromosome. Okay, which is circular in nature. Okay, which is all associated with the certain type of proteins. Okay, and this chromosome is looped and folded and attached and one to several point to the plasma membrane. Okay, because there is no nuclear membrane, so that's why it is reside inside the in the cytoplasm. Okay, so that's why it is looped and folded inside of the cytoplasm, and some point it is linked to the plasma membrane. Okay, and the DNA of E. coli, the most studied bacterial species which we know. Okay, it has the 4.6 million base pairs of 4.6 million base pairs, and it is around 10 means 1 millimeter long. Okay, yeah, then because the DNA is twisted and supercoiled, means to occupy the less space, it is twisted and highly supercoiled. Okay, and yeah, so the location of genes on a bacterial chromosome can be determined by the experiment of on the transfer of genes from one cell to another cell. Okay, so like this because it is highly supercoiled and there is a kind of transformation process from one cell to another cell, so we can determine easily the order of the gene. Okay, which is present in the DNA. Suppose this is the DNA, so through this natural process of transformation of one genetic material, one cell to another cell, we can actually identify the sequence of the gene. Okay, means which gene is firstly present, then after that which gene will come, like that we can identify through this transformation process. Okay, so yeah, so this is the there is like in naturally there are two type of the gene transfer method. One is the horizontal gene transfer and another one is the vertical gene transfer. Okay, so when the bacteria divide via the binary fission, then it will produce two offspring, right? So and then again in the third generation each cell will divide in again two cells, right? So this transfer from one cell to like another cell, or I can say like parent to offspring, when the gene mobility happens, then it is called the vertical gene transfer. Like in case of the human, when parents is giving their genetic information to their daughter cells, then it is called as the vertical gene transfer. But when two intra-species cell when transfer the genetic material to each other, then it is called the horizontal gene transfer, right? So like here in case of the horizontal gene transfer, here you can see that this is the donor cell and this is the recipient cell. Okay, most of the time it is intra-specific, but it can be also like inter-specific also. Okay, so they are not like parent and offspring, they are in the same generation, right? But the cell is different. Okay, means this is one cell and this is another cell, it is not the daughter of this cell, right? These are two individual cells of the same generation. Then it is when the transformation of genetic material happens between these two type of cell, then it is called the horizontal gene transfer. Okay, these are the two basic mechanism through which the genetic transfer happen. Okay, so the when the genetic transfer happen between means parent to offspring, then it is called as the vertical gene transfer, and when it is happen between two cells of the same generation, then it is called as the horizontal gene transfer. Okay. So, mechanism of genetic exchange of horizontal gene transfer is basically happen through basic pathway. One is the transformation, another one is the conjugation, and third one is the transduction. Okay. So, what is the transformation? Where the bacteria can acquire the DNA from means suppose this is the bacteria. Okay, two bacteria is present here. Okay, of the same generation. So when this bacterial DNA is occupied by this bacteria, okay, this is the suppose donor and this is the recipient. Okay, so it should for transformation it should have certain type of means capability to take up this DNA from this bacteria, right? So this capacity is called as the competency. Okay, competency. So this is the donor and this is the recipient should have the competency power, so that this when this bacteria will donate its DNA, then this recipient can take up this DNA. Okay, so that power is called as the competency, and this recipient cell is called as the competent cell. Okay, because it is competent to take this DNA. Okay, so that this process is called as the transformation, when the bacteria can acquire the foreign DNA inside of their chromosome. Okay, and that is the transformation. And second one is the conjugation. Okay, so conjugation is it is the uptake and retention of external DNA molecules. Okay, so in case of conjugation, between donor cell and recipient cell should be in contact with each other. Okay, then only conjugation will be possible, otherwise it is not possible. For that conjugation happen, it should be in close proximity to each other, so that it can produce the sex pili between them, so that the gene will be flow from this cell to this cell. Okay, so that is called as the conjugation. The third here yeah, this is the direct transfer of DNA from one bacteria to another bacteria, but for that it requires the close proximity to each other. Okay, and third one is the transduction. In case of transduction, the transfer of DNA between bacteria is mediated by the one virus, that is called the bacteriophage. Okay, so in that case what is happening, this is suppose bacteria, bacteriophage is the what I can say, it is the parasite for this host. Okay, so it will take the DNA from this host, and then it will transfer it into the another host bacterial cell. Okay, this bacteriophage will be transferred through this by taking the DNA of this cell to this cell. Okay, so these are the three different mechanism through which the genetic exchange happen. Okay, so the first one is the transformation. In transformation, what is happening, the recipient cell is competent enough to take the foreign DNA to inside of their cell. Okay, so this type of cell is called as the competent cell, and this by taking or acquiring this DNA, this capability is called as the competency. Okay, and this process from from transfer of one DNA to another DNA by using competency capability, that is called the transformation. Okay, the second one is the conjugation. In case of conjugation, there is the close proximity is required between two cells. Okay, and yeah, so that is the donor cell and this is the recipient cell. Okay, when they come close together, then they will create a one channel between them, so that this DNA can be transferred from donor to recipient. Okay, so this method is called as the conjugation. Okay, and this is the direct transfer of DNA from one bacteria to another bacteria. The third one is the transduction. So in case of transduction, there is the transfer of DNA between bacteria. Okay, so this is the two bacterial cell. It is mediated by the virus. Transduction means it is mediated by the virus. So virus is bacteriophage. Okay, so bacteriophage will firstly attack this donor cell and take the DNA from here, and it will transfer from here to here. Okay, So that process is called as the transduction. So we will see very like in summary wise. So what is the transformation? So suppose this is the donor cell. Okay, so donor cell, donor bacteria disintegrate and liberate its DNA, okay, DNA fragment into the surrounding environment. Okay, so firstly it will lysed and it will liberate its DNA outside of the cell. Okay, and because this recipient cell is like competent enough to take or carry this DNA, so that this DNA will come and bind to the surface of this recipient cell. Okay, through the help of DNA binding protein which is present in the recipient cell surface. Okay, it will bind here and it will then nuclear digestion of one DNA strand permit the other strand to enter the recipient cell. Okay, so what is happening here, the two strand when attached to the cell surface, only one strand will go inside. Okay, other strand will be digested on the surface of cell because of the nuclease activity. Okay, because it is free, so that's why it is degraded and only one strand will go inside. Okay, and this single stranded DNA now recombine with the homologous region of the bacterial chromosome. Okay, here you can see this red color. Now after going inside of the cell, what is happening, through the homologous region it is doing the homologous recombination. Okay, and this recipient cell is now called as the transformed because it is developed as new character inside of the cell, right? Because apart from its own characteristic, it also acquires some other kind of characteristic which is encoded by this part of the gene. Okay, so this is the process called as the transformation. Okay, so under right condition, bacteria can take in external DNA fragment by transformation, and DNA binding protein transfer external DNA across cell envelope. Then bacterial cell capable of transformation are referred as the competent cell. Okay, and homologous recombination can then occur. So this is the whole process of transformation, where what is happening, this is suppose the donor cell, when it is dead or when it is lysed, what is happening, the inside nucleic acid material will be released. Okay, so that time there is other type of cell which is called as the recipient cell or competent cell, which is having the capability to take this DNA and to like upgrade its genetic content. Okay, so this how it is doing that, so once it is released, this DNA will come and bind to its surface. Okay, and this is the DNA binding protein which will stabilize all this complex DNA and this cell complex. Okay, how it is coming to the surface, because of the charge. Okay, because there is the DNA is negatively charged, so there is certain type of proteins which is having the positive charge, so it will attract so that it will go and bind here in the cell surface. Okay, and once the means out of two strand, one strand will be cleaved by nuclease activity, so that only one strand will be remain here which will go inside. Okay, and after going into inside, it will integrated into the bacterial chromosome of the recipient cell through the homologous recombination. Okay, so and after integration, now this cell means cell is called as the transformant. Okay, because it is now adopted another type of the phenotype. Okay, which is not previously present in this cell. Okay, so now it is called as the transformant. Okay, and this process is called as the transformation. So for that Griffith, one scientist, done one experiment that is called the Griffith transformation experiment. Here what he did is it in mice, in the first group it gave pathogenic sensitive strain, and in the second group it gave the resistant strain. In the third group it killed the pathogenic strain. Okay, and then injected into the mice, and in the fourth group it is mixed the both the thing and then injected into the mice. So after a certain time, what he observed is colonies of the pathogenic cell isolated from dead mice. Okay. So because it was sensitive, so that's why when means when we like yeah, when Griffith injected the sensitive strain, that is encapsulated pathogenic cells into the mouse, it developed the pneumonia and it is died. Okay. And in this group, what is happening when they injected the resistant strain which is uncapsulated. Okay. For means bacteria or virus, you maybe know that there is the encapsulation process where they like cover its infective materials inside. Okay. So in that case, in the resistance strain, what is happening, there is no encapsulation and it is harmless. So that's why there is no harm to the mice, and now mice is survived because they are unable to infect the disease. Okay. And the third one is the heat killed pathogenic cells. Okay. So now what he did is further an injection of heat killed S strain, the sensitive strain, he heat killed, so that this particular encapsulation will be inactivated. Okay. And so that it it will be like it will be like normal only, because the encapsulation process has gone because of the heat killing. Okay. So that's why here also the uh, yeah, the cells will be furthermore injection of heat kill cells did not no harm because the cells were dead. Okay. Here because it was the heat inactivated, so that's why certain particle were remain like that only, so that it is cause disease, and it is died. Okay. And in the mix one, but when Griffith injected a mixture of live and heat killed heat sensitive strain cells into the mouse, it died. When because Griffith cultivated the organism from the blood, and he found that live this as strain cells was there. Okay. Because in case in these two cases, what is happening is, here the sensitive strain is not uh, completely gone, right? On when it is getting the what I can say, it is getting the proper time to grow, that time it is appearing and causing the disease, so that the colony is not isolating. Okay. But when it is mixing, then also it is having two things, right? Harmless as well as the pathogenic cells, so that it is like it is gaining it is transferring the DNA material to the harmless R strain, so that it will survive. So from here, this experiment, it is proved that the DNA is exchanged from one cell to another cell. Okay. So that is the transformation experiment. So in case of as I earlier told you, in bacteria there is two type of chromosome. One is the its original chromosome. Okay. Which is larger in size, and there is another extra chromosomal DNA. Okay. So that is called as the plasmid. Okay. So this is the extra chromosomal genetic elements. So the main genetic information will be inside the bacterial chromosome, but the more advantageous characteristic is present in the plasmid. Okay. So plasmid is what, like suppose certain bacteria is resistant toward any kind of the antibiotics. Okay. So that resistance characteristic is found in the plasmid. Okay. So that is not very much essential for the survival of this cell. It is providing the extra characteristic to the bacteria. Okay. So that is the that type of characteristic is present in the plasmid. Okay. And it is autonomously replicating unit. Okay. And it is means in bacteria, it is circular DNA. And as I told you, like it don't encode the essential function. Okay, only additional genetic information it will be coded, like it provides the resistance, okay, and toxin, protein, metabolizing properties. Okay, so the additional feature it will provide, it is not coding any kind of the properties which is essential for the bacterial survival. It is not encoding those things. Okay, it is only encoding the extra characteristic. Okay, and this plasmid are like two types. One is the F plasmid, another one is the resistance plasmid. Okay, so this F plasmid is required for the transformation of one gene to another cell. Okay, and this resistance gene, this resistance plasmid provide, as I told you, like antibiotic resistance, like that it this plasmid will provide. Okay, yeah, and this plasmid can be transferred through conjugation, transduction, and incorporation. Okay, and if we will see the classification of plasmid, we can classify into two categories. One is transfer properties, another one is the phenotypic properties. Okay, so the transfer properties is having conjugating plasmid and non-conjugating plasmid. Okay, so conjugative plasmid are those that mediated the conjugation, and these plasmid are usually large and have all the genes necessary for autonomous replication and for transfer of DNA to a recipient. Okay, it is having the genes for like conjugation tube or sex pilus. Okay, that is called as the conjugative plasmid. Non-conjugative plasmid is are those that can't mediate the conjugation, and they require usually smaller than the conjugative plasmid, and they lack one or more gene that is needed for the transfer of DNA. Okay, it is not self transferable, but it is the conjugating plasmid is self transferable. Okay, and it requires certain type of the this non-conjugating plasmid requires certain type of genes which it acquires from other plasmids. Okay, it is not present in those plasmids. Okay, so this is the conjugating and non-conjugating plasmid. Conjugating plasmid is self-sufficient to transfer, but this non-conjugative plasmid is not self-sufficient to transfer. Okay, then second one is the phenotypic effect. Based on the phenotypic effect, so like there are three type of plasmid. One is the fertility plasmid, okay, which is which can do the transfer of genetic information from one cell to another cell, that is called the F factor. Another one is the bacteriocynogenic plasmid, that is this plasmid have genes which encode the substances that kill other bacteria. Okay, these substances are called bacteriocins or colicins. Okay, so it is having the extra, like if suppose this is the bacteria and it is having the bacteriocynogenic plasmid, it means it will produce certain type of the substrate which will kill the other bacteria. Okay, it is providing the like extra features to this bacteria, right? It is not necessary for the survival of this bacteria, but it is giving its the extra advantage so that it can kill other type of bacteria. Okay, and another one is the resistance plasmid, which is like it is provide any kind of the resistance toward any type of antibiotic or any type of media, I can say. Okay, so these are the classification of plasmid. So now the bacterial conjugation. Okay, so in bacterial conjugation, what is happening is it should be like conjugative plasmid. Okay, this conjugative plasmid is the F factor. Okay, where this F factor is present, that is called the F positive cell or the donor cell. Okay, and the recipient cell which is going to take up this DNA, that is called as the like F negative cell or the recipient cell. Okay, and it should be in close proximity so that the conjugation can be happen. Okay, so once this close proximity happen, what is happening is now this F factor is having several genes which will mediate this transfer of F factor DNA to one cell to another cell. Okay, so some of gene will produce this conjugating tube, and some of gene will be helping in the integration of this F factor into this another cell chromosome. Okay, so replication and transfer of F factor once started from the ori T site. Okay, from where the transfer is happen, that is called the ORT site, origin of transfer. Okay, so from here the transfer process is started. Okay, and once this complete or I can say it is simultaneously, once this transfer is happen, it is simultaneously replicating the other strand of this F factor in the donor cell. Okay, one strand is transferring, another strand is simultaneously replicating here. Okay, so now both the cell will be having the same type of DNA content, so that's why both the cell will be F positive cells. Okay, it is also having F factor, it is also having the F factor. Okay, so yeah, and after this is happening, once this cell get integrated into this bacterial chromosome, then this F cell is called as the HFR, high fidelity transfer cell. Okay, so this process require an F plasmid, yeah, and has all conjugation gene in the F plasmid, and it direct the formation of sex pilus, and single stranded DNA produced by the DNA replication is transferred to F cell through the sex pilus. Recipient produce two strand. Okay, so yeah, this is the whole summary of the conjugation, where firstly this for happening conjugation, the bacteria should be having the F factor, only then only it can be transferred the genetic information. Okay, so this F factor this F factor, how it is having two classes of gene. Some of the gene will produce this conjugating tube, and another set of the gene will help for the transfer. Okay, so once where the site where the transfer started, that site is called as the origin of transfer. Okay, so from here this DNA strand will be transferred simultaneously in the donor cell. The another strand will be replicating. Okay, so once this strand reach to the recipient cell, it also make the another copy of this strand. Okay, and it can be reside like that only, means in separated way, or it can be integrated to the bacterial chromosome. So once it is integrated with the bacterial chromosome, then it is called the HFR. Okay, HFR cell, or if it is present as a separate unit, then it is called as the F positive cell. Okay, so like that, here the conjugation tube will be formed. Okay, because of the presence of F factor. So episome is also nothing but it is the kind of F factor which is having the replicating unit. Okay, it is the portion of genetic material that can exist independent of the main body of genetic material at some time, while at other time it is able to integrate to the chromosome, like F factor, which can be able to present as a separate unit, or it can be integrated to the main bacterial chromosome. Okay, so episome, we can consider the F factor is also a kind of episome. Okay, and yeah, so when it is integrated, it is called as the HFR strand. Okay, which is having the high frequency of recombination, because it exhibit a high efficiency of chromosomal gene transfer in comparison with F cell. Okay, so here F positive cell, the recombination frequency will be increased when it is integrated with the main chromosomal DNA. Okay, comparative to this F cell. Okay, so yeah, that is the episode. So here how HFR conjugation happen, that we see. Okay, if the F factor plasmid inserted into the host chromosome, then it is called as the HFR cell, and this will result in the transfer of the entire DNA complex. Okay, so suppose this is the F positive cell, recombination between the F factor and chromosome occur at a specific site on each. Okay, So because of the homologous recombination, it will integrate, and after integration, it is called as the HFR. Okay. And when the F factor become integrated into the chromosome of F positive cell, it makes a cell of high frequency recombination cell. Okay. So when now you are having the HFR cell. Okay. So when you are conjugating the HFR cell to recipient cell which is not having the F factor, then what is happening? Then the final recombination will give you the HFR cell and the recombinant F cell. Okay. This probability you have to remember, because in exam point of view it can be asked, like when one F cell, F positive and F negative will cross, what conjugate, what will be the offspring. Okay. Not offspring, what type of cell will be generated, like that when HFR will conjugate with F negative cell, what will be the result. Okay. So it will be the HFR cell and the recombinant F negative cell only. Why? Because when it is transferring the HFR cell to recipient cell which is not having the F factor, it takes time. Okay. So and before once it is completely transferred, then only we can say either it will be F positive cell or HFR cell. Okay. But before completing this process, this conjugation tube will be breaks. That's why it is remained as a HFR cell and the recombinant F negative cell. Why it is called as recombinant cell? Because some of the part of HFR cell is already transferred but not completely. If it is completely transferred, then it is called as the either F positive cell, because if it is present as a separate unit, then it is called as the F positive cell. And if it is completely transferred and integrated, then it will make the HFR cell again. Okay. But because transfer process takes time, so that it is only some part is only transferable, so that it is remain as a HFR cell and a recombinant F cell. Okay. And this conjugation thing we can use for the transformation, we can use for the genetic mapping, right? So how we will do the genetic mapping? Suppose this is the bacterial cell. Okay. And if the plasmid is having 2-3 genes, like A, B, C, like that genes is having, so we can while transferring, if it is broken in the mid of the process, then if suppose A is close to B, then in different bacterial cell, some of the population will be, and most of the population will be having the AB gene, right? And only some of the population will be having the C gene. Okay. Or A gene, or individual B gene. Okay. But most of the population will be having the A and B gene, so that we can say it is transforming, so that it means they are near to each other, then only they are transferring together. Okay. So like that we can map the gene sequence also. Then the third process is the transduction. Okay. So what is the transduction? So as I told you, when the transformation process is mediated by the bacteriophage or the virus, then it is called the transduction. Okay. So a virus particle can transfer DNA fragment from one host cell to another cell, followed by the recombination. Okay. So suppose this is the yeah, this is the first step na. So this is the bacterial chromosome or the donor cell. Okay. And one phase particle infect this bacteria. Okay. And then phage DNA and protein are made, and the bacterial chromosome is broken into pieces. Okay. And now what will happen is, occasionally during the phase assembly, means now phase will multiply inside the host cell. Okay. And it will be encapsulated inside it will be encapsulated during the packaging of the virus. Okay. And the donor cell lysed and released phase particle containing the bacterial DNA. Okay. So after multiplying, what is happening? Each content here whatever DNA is present, it will be go each of the bacteriophage. Okay, and once it is lysed, then this each bacteriophage can go and infect to the other type of bacterial cell. Okay, so that time while infecting, what is happening is that time it is also transferring the genetic material of one bacteria to another bacteria, that is called the recipient cell, which is like acquiring this genetic material. Okay, phase carrying bacterial DNA infect a new host cell, the recipient cell. Okay, and last one is recombination can occur, producing a recombinant cell with a genotype different from both donor and recipient cell. Okay, so this is the transduction process. So what is happening, like suppose this is one bacteria which is called as the donor cell. Okay, one bacteriophage come and attack or infect this bacteria. Okay, and after attacking or after infection, what is happening is it is degrading the bacterial chromosome of the donor cell. Okay, and once it is degraded, it is multiplying inside of this donor cell. The bacteriophage is multiplying. Okay, it is increasing the number of itself. Okay, and once it is reached to the sufficient number, then it has to like burst. Okay, it has to burst. So that time what is happening is, before that like as a complete virus unit, what it needs, it should be encapsulated. Okay, the it should be require proper assembly. So while proper assembly, what is happening is, with its own DNA, it is also packaging it the bacterial DNA. Okay, so some of the bacteria will be having the mix of both the DNA, some of will be like having only the phage DNA, some of will be having only the like this bacterial DNA. Okay, so when it will burst, it will release the bacteriophage. Okay, and this bacteriophage will go and affect to the another recipient, another cell, which is called as the recipient cell, because now this bacteriophage is transferring its another bacterial stuff into this cell, right? So that's why through that way the transformation happen of or we can say like genetic exchange will happen. Okay, and once go inside of this recipient cell, what is happening, it can be recombined with the recipient cell chromosome. Okay, through the like recombination, homologous recombination mechanism. Okay, so this complete process is called as the transduction. So through that also the like genetic mapping happen. Okay, like conjugation, this is the conjugation. So because here HFR strain is there. Okay, so HFR strain produced in case of the like conjugation. Okay, so how we can do the mapping? So suppose there is a different type of HFR strains. Okay, so it is like that the HFR strain transfer starting site can be different. Okay, it depends on the strain. Like here H this is the H strain, so it is transferring starting site is the threonine. The one is having the transfer site is the leucine, two is having the proline, and this is the some tone gene is there. Okay, so everyone is like starting from the origin of transfer, but the sequence is can be different. Okay, in all the cases, but how you will know that which means the genetic information, the sequence of the genetic information, right? So for that what you will see is firstly check here, like it is in case of H, it is threonine, leucine, Ag, tone, proline, lacgene, galgene, and threonine. Okay, so it is finished in the like threonine something kind of the gene. Okay, so in the second strand you have to look like after threonine what gene will come. Okay, so here if you will see it is like Ag, tone, proline, lac, galactine, and then again threonine. Okay, so it is repeat. Here it is in this way. Here it is in this way. Okay, so after threonine, what is coming? Threonine and leucine. So the sequence will be what is firstly threonine, then it is circular. Okay, so firstly threonine, then leucine, then AG gene, then tone gene, then proline gene, then lactin gene, then galactin gene, okay, then this Thi gene, and after Thi gene there is threonine, okay, and then leucine. Okay, now you have to see where is in the third strain this sequence is matching or not. Like here threonine after means it is again coming from this side, right? So till leucine we have mapped. Okay, so till leucine we have mapped, again it is coming from the this AG gene. Okay, so yeah, uh, AG gene. So it is not after that it is tone gene, na? Yeah, but how come it is AG gene? Yeah, again here AG will come, okay, then tone will come, then proline will come, okay. So here it is the sequence, if you will see, okay. After proline there is lac, again gal, then threonine, thegene, threonine, leucine, agene and tone, okay. Then it will be like that, this sequence, okay. So like that you have to map. So firstly you have to check like in first strain what is the sequence, okay, and after that in remaining strain you have to match the sequence. Like in, if here it is thegene is the lastest one, then here after thegene what is coming, threonine and leucine, okay. So means after thegene there will be like threonine and leucine, and from the third strain you can find out if further there is any gene or not. So thegene, then threonine, then leucine, then again it is having the AG and tone gene and proline, okay. So you have to write down that gene proline, no, it will be like leucine will be the last one, okay, because it is again coming from this side, okay. This is like coming the same side, so that's why it is the same sequence. If you will see here, it every threonine, leucine, agene, tone, proline, lactin, galactin, threonine, okay. So like that you can map everyone, the sequence is same but the starting of transfer is different, okay. So from there you can identify like what is the sequence of these genes, okay. So yeah, so like in case of the transduction, it is the transfer of DNA from one bacteria to another via bacteriophage, and bacteriophage is a virus that can attack the bacterial cell, and after that the bacteria can undergo two type of the cycles. One is the lytic cycle, another one is the lysogenic cycle, okay. And transduction is two type, one is the generalized transduction and another one is the specialized transduction. So in case of generalized transduction, the fragment of the bacterial cell is having equal opportunity to means package inside of the bacteriophage, okay. It is having if all the fragment of donor DNA from any region of chromosome have a chance to enter into transducing bacteriophage, then it is known as the generalized transduction, okay. Every fragment is having the equal chance to transfer, okay. In this type of transduction, at first bacteriophage infect the donor cell and begin its lytic cycle, okay, because only then only it can be like infect the other cell. If it sustain into the lysogenic cycle, it won't be able to like transfer the DNA from one cell to another. So for that it should be like process the lytic cycle, okay. And this is the normal transduction process. Once enter, it will degrade the bacterial chromosome and package inside of every bacteriophage, and then lytic cycle will happen, and each bacteriophage can go and transfer the genetic material to other cell, okay. So that is called as the generalized transduction. Another type of the transduction is the specialized transduction. So in case of specialized transduction, bacteriophage transfer only a few restricted gene, okay, from donor bacteria to recipient bacteria, and this is and this type of bacteria is also specialized because it is happened with the help of temperate bacteriophage, okay, which undergoes lysogenic cycle in donor cell, okay. At first temperate bacteriophage enter into the donor bacteria, and then its genome gets integrated with host cell DNA at certain location, okay, and remain dormant and pass generation to generation into daughter cell during cell division. The bacteriophage which follow lysogenic cycle is known as the temperate phase, okay. So bacteriophage are also different types, okay. So one of the bacteriophage is the temperate phase which follows the lysogenic cycle, okay. When such lysogenic cycle is exposed to certain stimulus such as the some chemical or UV light, it cause the induction of virus genome, and it cause the begins of the lytic cycle, okay. On induction from donor DNA, this phage genome sometimes carry a part of the bacterial DNA with it, and the bacterial DNA lies on side the integrated phage DNA are only carried during the induction. When such bacteriophage carries a part of donor bacterial DNA infect a new bacteria, it can transfer that donor DNA fragment into new recipient cell. So in this specialized transaction, only those restricted genes are situated on side of the integrated viral genome have a chance to enter into the recipient cell, okay. So in case of like specialized transduction, what is happening is firstly the temperate phase will attack, okay, or infect the donor cell, and it will its DNA will go and not degrade the bacterial cell, instead degrading it will go and integrated into the like bacterial chromosome, okay, and it will form like this, okay, and replicon of viral DNA and once it get the stimulus, what it will do is it will proceed for the lytic phase. In that time what is happening is only some part of the DNA which is integrated into the virus DNA, that will package inside of the bacteriophage, okay, and it will burst, and when it will go to the recipient cell, only the part of the bacterial chromosome will be like infect or transfer to the recipient cell, okay. So that is the specialized transduction, okay. So in specialized transduction, both the mechanism is followed, firstly the lysogenic phase, then lysogenic phase is the dormant phase, okay, and it will pass, it can passes like generation to generation, and once it get stimulus it can converted into the lytic phase, okay. But in case of the generalized transduction, from starting itself it will proceed the lytic phase, okay, and each fragment of the cell is having equal capacity to like transfer from one cell to another cell, okay. So this is two different type of the means procedure for the transduction, okay. So these are the three different approaches which is like helpful for the transfer of one transfer of genetic material from one cell to another cell, okay. So regarding this, one question has been asked: in nature the horizontal gene transfer across bacteria is mediated by gene cloning followed by transformation, conjugation and transformation, conjugation only, or transformation only. What will be the correct answer? B will be the correct answer, okay. Conjugation and transformation, because horizontal gene transfer happens between the two species of same generation, and it can be happen either via transformation, either via conjugation, or the like transduction, okay. So here in the option only conjugation and transformation is given, so this B will be the correct option. Only conjugation is not correct answer, and only transformation is not correct answer, because it is happening through three different ways, okay. So yeah, this is the correct answer: conjugation and transformation.